Today we'll be learning about RNA, which comes from DNA, which is used to specify different proteins. There are two steps to this process: transcription and translation.
We're going to talk about transcription. It all starts with messenger RNA, which is a type of RNA synthesized using a DNA template that attaches to ribosomes in the cytoplasm and specifies the primary structure of proteins. RNA is similar to DNA, but RNA is single-stranded, while DNA is double-stranded. DNA looks like this. It's a double helix.
RNA looks like this. It's a single strand.
At the end of transcription, one new RNA transcript will be completed. The process of transcription begins when RNA polymerase pulls apart the two strands of DNA and joins RNA nucleotides as they base pair along the DNA template.
RNA polymerase only works in the 5' to 3' direction. Remember, the 5' end has a phosphate group and the 3' end has a hydroxyl group. RNA polymerase doesn't need a primer.
The 5' end has a phosphate group and the 3' end has a hydroxyl group. Well, how does RNA know where to start transcription on the DNA template?
Well, there is something called the promoter, which is a specific sequence of nucleotides along the DNA, marking where transcription of a gene begins and ends. In bacteria, the sequence that signals the end of transcription is called the terminator. Think of the direction of transcription as going downstream from the promoter to the terminator. Upstream is 5' to 3', and downstream is 3' to 5'. The stretch of DNA that is transcribed into an RNA molecule is called a transcription unit.
There are a couple differences between transcription in bacteria and transcription in eukaryotes. Bacteria have a single type of RNA polymerase that synthesizes mRNA and other types of RNA that function in protein synthesis, such as ribosomal RNA. For further reference, ribosomal RNA, or rRNA, is the most abundant type of RNA which, together with proteins, make up ribosomes. A ribosome is a complex of RNA and protein molecules that functions as a site of protein synthesis in the cytoplasm and consists of large and small subunits. In eukaryotic cells, each subunit is assembled in the nucleolus.
In contrast, eukaryotes have at least 3 types of RNA polymerase in their nuclei. The type of RNA polymerase used for mRNA synthesis is RNA polymerase II. Here is a picture of RNA polymerase II, a very important enzyme in transcription. Other RNA polymerases transcribe RNA molecules that are not translated into proteins. Also, the details of termination differ between bacteria and eukaryotic cells. A final difference is that in a bacterium, the RNA transcript is immediately usable as mRNA. But in a eukaryote, the RNA transcript must undergo processing, which we will describe later. There are 3 steps of transcription: initiation, elongation, and termination. Let's start with initiation. The promoter which we mentioned earlier, which is made out of nucleotides, extends a few dozen nucleotide pairs from the start point. The promoter determines which strand of the DNA, double helix, will be used as a template. In bacteria, the RNA polymerase itself recognizes and binds to the promoter. In eukaryotic cells, a collection of proteins called transcription factors mediate the binding of RNA polymerase and the initiation of transcription. RNA polymerase II will work when certain transcription factors are attached to the promoter. Acetylation of histone tails promotes loose chromatin structure, which promotes transcription.
The complex of transcription factors and RNA polymerase II bound to the promoter is called transcription initiation complex. The Tata box is a crucial DNA promoter sequence for forming the initiation complex at a eukaryotic promoter. Let's go into further detail about the Tata box. This is the Tata box. It starts with thymine, adenine, thymine, adenine, and then 3 more adenine. On the 3' end, it has the corresponding base pairs, so adenine, thymine, adenine, thymine, and 3-thymine. A eukaryotic promoter commonly includes a TATA box. TATA stands for thymine, adenine, thymine, adenine, the nitrogenous bases. Several transcription factors must recognize the TATA box and bind to the DNA before RNA polymerase II can do so. Finally, other transcription factors bind to the DNA along with DNA polymerase II, and then the DNA is unwound and RNA synthesis begins. As you can see, protein-protein interaction is important in transcription.
The next step is elongation. In elongation, RNA polymerase continues to untwist the DNA double helix. As it does so, it exposes about 10 to 20 DNA bases at a time for pairing with RNA nucleotides. RNA polymerase adds nucleotides to the 3' end of the growing RNA molecule. This would be in the upstream direction. Because the RNA is growing with new nucleotides, the RNA transcript molecule peels away from its DNA template and the DNA double helix reforms. Transcription progresses at a rate of about 40 nucleotides per second in eukaryotes. More than one RNA polymerase molecule can transcribe a single gene at a time. A growing strand of RNA trails off from each polymerase, with the length of each new strand reflecting how far along the template the enzyme has traveled from the start point. The congregation of many polymerase molecules simultaneously transcribing a single gene increases the amount of mRNA transcribed from it. This helps the cell make the encoded protein in large amounts.
The next step in transcription is termination. As we mentioned earlier, termination is different in eukaryotes and bacteria. In bacteria, transcription proceeds through a terminator sequence in the DNA. The transcribed terminator functions as the termination signal, causing the polymerase to detach from the DNA and release the transcript, which is available for immediate use as mRNA. In eukaryotes, RNA polymerase II transcribes a sequence on the DNA called the polyadenylation signal sequence. The first step of transcription is initiation. After the RNA polymerase binds to the promoter, the DNA strand unwinds and the polymerase initiates RNA synthesis at the start point on the template strand. The second step of transcription is elongation. The polymerase moves downstream, unwinding the DNA and elongating the RNA transcript, 5' to 3'. In the wake of transcription, the DNA strand reforms a double helix. The third step in transcription is termination. The RNA transcript becomes long. Eventually, the RNA polymerase detaches from the DNA strand. The result is a completed RNA transcript. Now, we are going to talk about how eukaryotic cells modify RNA after transcription. Enzymes in the eukaryotic nucleus modify pre-mRNA in specific ways before the genetic messages are dispatched to the cytoplasm. This is called RNA processing. Both ends of the primary transcript are altered. Also, during RNA processing, some interior sections of the RNA molecule are cut out and the remaining parts are spliced together. The slight modifications produce an mRNA molecule that is ready for translation.
The two ends of a pre-mRNA molecule are each modified in a particular way. The 5' end is synthesized first by receiving a 5' cap. A 5' cap is a modified form of a guanine nucleotide added onto a 5' end after transcription of the first 20 to 40 nucleotides. The 5' cap is also called a guanine cap as it consists of guanine. The 3' end of the pre-mRNA molecule is also modified before the mRNA exits the nucleus. At the 3' end, an enzyme adds 50 to 250 more adenine nucleotides, forming a poly-A tail. The 5' cap and the poly-A tail both serve important functions. First, they help export the mRNA out of the nucleus. Second, they help protect the mRNA from degradation by hydrolytic enzymes. Finally, the 5' cap and poly A tail help ribosomes attach to the 5' end of the mRNA once the mRNA reaches the cytoplasm. Here's a picture of the 5' cap and poly A tail on the completed RNA transcript. At this end is the 5' cap. A modified guanine nucleotide is added to the 5' end. Here is the protein coding segment. This includes the start codon and stop codon. Finally is the poly A tail. (50 to 250) adenine nucleotides are added to the 3' end.
The RNA transcript molecule is very long, about 27,000 base pairs long. However, it only takes 1,200 nucleotides to code for an average size protein of 400 amino acids. Remember, each amino acid is encoded by a triplet of nucleotides. This triplet follows the triplet code, which is the genetic instructions for a polypeptide chain that is written in the DNA as a series of non-overlapping 3 nucleotide words. Each mRNA base triplet is called the codon.
The following is an illustration of the triplet code. The genetic instructions for the polypeptide chain are written in a series of 3 nucleotide words called codons.
This means that most eukaryotic genes have long stretches of non-coding nucleotides. RNA splicing is used to remove the non-coding portions of the RNA transcript. The non-coding segments are called introns. In contrast, the coding regions are called exons because they are eventually expressed into amino acid sequences. As well as the mRNA being altered at both ends, it also goes through RNA splicing. RNA splicing, specific to eukaryotic cells, is the removal of introns.
After the introns are removed, the remaining exons are spliced together, forming an mRNA molecule with a continuous coding sequence. Before RNA splicing happens, the RNA transcript is called pre-mRNA, and after it is spliced, it is called mRNA.
At each end of an intron, there's a short nucleotide sequence called small nuclear ribonucleoprotein, SNRPs, that recognize the splice sites. SNRPs are composed of RNA and protein molecules. The RNA in a SNRP particle is called a small nuclear RNA. Each RNA molecule is about 150 nucleotides long.
Several different SNRPs join with additional proteins to form an even larger assembly called a spliceosome. The spliceosome interacts with certain sites along an intron, releasing the intron and joining together the 2 exons that were on either end of the intron. Small nuclear ribonucleoproteins, SNRPs, and other proteins form a molecular complex called a spliceosome on a pre-mRNA molecule containing exons and introns. Within the spliceosome, snRNA base pairs with nucleotides at specific sites along the intron. The spliceosome cuts the pre-mRNA, releasing the intron, and at the same time splices the exon together. The spliceosome then comes apart, releasing mRNA, which now contains only exons.
Important enzymes in RNA splicing are ribozymes. Ribozymes are RNA molecules that function as enzymes catalyzing reactions during RNA splicing.
Now that we have touched on all the aspects of transcription, let's make an evolutionary connection. This has to do with genetic variation, and nothing to do with common ancestry. Kara! An interesting concept relating to evolution in terms of transcription is the function of introns. An interesting concept relating to evolution in terms of the presence of introns in genes is that a single gene can encode more than one kind of polypeptide. Many genes are known to give rise to two or more different polypeptides, depending on which segments are treated as exons during RNA processing, a process called alternative RNA splicing.
The presence of introns in a gene may facilitate the evolution of new useful proteins as a result of a process known as exon shuffling. Introns increase the probability of potentially beneficial crossing over between the exons of alleles. Exon shuffling could lead to new proteins with different functions.
Let's review that section of information. Question 1. How does the alteration of the 5' and 3' ends of pre-mRNA affect the mRNA that exits the nucleus? The answer is, the 5' cap and poly A tail facilitate mRNA export from the nucleus. Also, it prevents the mRNA from being degraded by hydrolytic enzymes and facilitates ribosome attachment. Question 2. How is RNA splicing similar to editing a video? The answer is, when editing a video, segments are cut out and discarded, just like introns are in RNA splicing. Then, the remaining segments are joined together like exons so that the regions of joining are not noticeable.